do. I have them all, but they are all back in my office. I'm heading there right after class. You'd be welcome to follow me over there, or I'll be in my office between 2 and 4 this afternoon. I don't know if you saw my Quirkus announcement, and I'll just be handing out essays. So either one would be great. Perfect. I'll see you there. Thank you. Hello. Give me 30 seconds to just take this microphone off. Here we are. Okay. I will leave the charge out.
Okay, all right, good afternoon, everyone. I hope you had a good Thanksgiving holiday weekend. A couple of housekeeping things before we dive into the lecture this afternoon. So one is to say that, of course, the rebuttals for Term Quiz 2 I have not yet had a chance to look at. That's mostly a product of the Thanksgiving weekend, so it's on my to-do list for tomorrow, so hopefully if you submitted a rebuttal, you'll get a response by tomorrow. We have Term Quiz 3 coming up next Monday and Tuesday. So that focuses on the stuff we've covered over the last couple of weeks. So yeah, the practice quiz questions for last week are posted already, so that's going to give you an opportunity to get practiced up for that. Not that we seem to need much practice. Term Quiz 2, a little worse than Term Quiz 1, but still an 80 average. So I tried harder, I thought, and you guys kept up. So I'll keep trying harder and see if you can keep keeping up, and that'd be great. Yeah, and that's all I was going to say, I think. Any questions about things before we continue on with the lecture here this afternoon? Everybody's all good? Okay. All right, so we were talking about graded potentials at the end of class on Friday, and the last thing about them that we had discussed was this idea of decremental conduction. So basically the idea that when you have a graded potential initiated somewhere by a stimulus, you're going to have a graded potential occur right in the area where the stimulus occurred. But that graded potential is not going to stay put. It's going to spread from that location in both directions, okay? And we said that's because in the case of this depolarization that we saw here, those positive charges are attracted to the negative charges that they see in both directions here in the inside of the cytosol, and so they start moving in both directions, and therefore making the adjacent regions more positive as well, so causing graded potentials to occur in both directions from the site of the stimulus. And then we noted that as these positive charges that were on the outside of the membrane, came in, as we said, through this open gated channel here, this region becomes more negative. So these adjacent positive charges on the outside of the membrane will slide over to replace the positive charges that were once here, and that means you'll get negative charges developing on the outside of the neuronal membrane as well. And so these positive ions that are inside, they'll have two choices. They'll either continue to travel inside the cytosol, attracted to the negative charges there, or they will cross over the membrane to get to the negative charges that they sense developing on the other side of the membrane. And it's that crossing back over the membrane of those charges that ultimately leads to the decremental part of decremental conduction, because as those positive charged ions move back across to the outside, they are returning the membrane potential back to its original resting state. So that was decremental conduction and what's driving that. We ended with this figure here very hastily at the end, so I'll just kind of cover it again here. In this figure, we have a stimulus that is occurring right here, so at this region of the cell body, kind of on the other end from where the axon initial segment is. So here they're using this kind of cuter term, I guess, this trigger zone, to refer to the axon initial segment. And of course, what they're trying to get at there is that that's the region of the neuron where the concept of the threshold potential matters, right? The idea here is that, graded potentials might be able to get us to threshold potential, and if they do, then an action potential occurs in response. And so this axon initial segment is the first part of the neuron that has the capacity to generate an action potential. So it's the graded potentials that are coming into that region, the axon initial segment. If they get us to the threshold potential or above, then an action potential will occur. So here we have this stimulus at the very far end of this cell body, of this neuron. And you can see they've got a little recording electrode in here to measure the membrane potential in this region. And you can see that in response to the stimulus, we've had a depolarization occur. And you can even see that it has gone above the threshold potential. right? Now, the concept of threshold potential means nothing over here in this region of the cell body. Again, it only matters here. But we just note that it was a supra-threshold stimulus. It got us above the threshold potential out there in that distal region where the stimulus occurred. We then noticed that by the time the gradient potential got to the center of the cell body, where their second electrode was, you could see that it had decayed, so it was just hitting the threshold potential now. So a threshold at this point. But by the time it's actually traveled to the axon initial segment, where the threshold potential matters, further decay of that gradient potential has led it to become a subthreshold potential. So as they say here, we get no action potential in this case in response to the stimulus, because although the stimulus was suprathreshold when it was first generated, out there at the opposite end of the neuron, by the time it had conducted itself to the axon initial segment, it had decayed to such a point that it was now a subthreshold stimulus, not capable of generating an action potential. Okay? So that's one of the things that is significant about decremental conduction. So then, of course, with this figure in mind, you might say to yourself, well, given this concept of decremental conduction, how is it even possible, therefore, that a stimulus could ever cause an action potential to occur? If the gradient potential that results from that stimulus is just going to decay as it travels through the cell body and becomes subthreshold by the time it gets to the axon initial segment, how could a stimulus ever cause an action potential to occur? Well, there are two answers to that question. Obvious answer number one is that if this stimulus here had been stronger, right, then we would have gotten a greater magnitude for our gradient potential. And perhaps then, by the time it had decayed and gotten to the axon initial segment, even though it would have decayed, it would still be strong enough to actually trigger an action potential. So a stronger stimulus, notwithstanding the decremental conduction, could still be strong enough when it gets to the axon initial segment to generate an action potential. So that's one answer. But answer number two is to realize, of course, that there is very seldom going to be just a single stimulus acting on a neuron, and therefore it's unlikely that we'll have just a single gradient potential that's being formed and traveling to the axon initial segment. We discussed earlier in the course that a single neuron can be interacting with 60,000 other neurons. So the chances are a given neuron is probably receiving many stimuli at the same time, forming many gradient potentials, and it's the summation of all those individual gradient potentials at the axon initial segment that actually can get you also above the threshold. So that leads us then to this conversation about summation and cancellation here next. So I'm going to walk through the figures here. I'm going to do summation first, so the two right-hand figures, and we'll start on in the center, I guess, with spatial summation. So what we're looking at here is they are measuring here the membrane potential of this orange neuron here. Okay, you can see its resting potential right there. You can see the threshold potential is marked out with the dashed line. And what they're showing us here is that there are these two other neurons, right, that form synapses with this orange neuron. And it tells us that they are both excitatory. The positive symbols there are meant to tell us they're excitatory, which means they're going to cause a depolarization of the membrane potential of that orange neuron, bring it closer to threshold, increase its likelihood of forming an action potential. But what they're also trying to illustrate for us here is that if we had just this red neuron's excitatory input here, right, you can see that it causes a depolarization to occur, but does not get us to the threshold potential. So the excitation of this one red neuron is not sufficient to get the orange neuron up to a threshold potential. And similarly, what they're trying to show us here too is that the purple neuron also on its own only causes a subthreshold gradient potential to occur. So neither of these two neurons can excite the orange neuron enough to create a gradient potential in that orange neuron that would get you to threshold and cause an action potential to occur. However, if the red and purple neurons together are both simultaneously exciting the orange neuron, then what they're trying to show us here is their two depolarizations add together. They sum together to get us to the threshold, and that's why this orange neuron is showing an action potential. So this is spatial summation because, as it says here at the bottom, you have excitatory potentials coming from many different neurons into this one neuron, this orange neuron, and the excitatory potentials, the depolarizations, these depolarizing gradient potentials from these multiple neurons are summing together to get us to the threshold potential where an action potential then occurs in response. Okay, so that's spatial summation. On the right, we have temporal summation. So here again, we have our orange neuron whose membrane potential is being measured in the figure here. And in this case, there's only a single neuron that is forming a synapse with it, this green neuron. And so what they're showing us again here is that the green neuron basically is kind of sending two excitatory stimuli to this orange neuron. You can see the first one coming here, right, again is subthreshold, does not get the orange neuron to its threshold potential. And the second one is similar, again on its own, subthreshold, and can't get us to the threshold potential. But the fact that these two come kind of right behind each other, one then the next, means that the first one has caused a depolarization that's still present when the second excitatory input comes in. And so now we have these two depolarizations, one after the other, both adding together to get us to the threshold. So here we have temporal summation now because it's only a single neuron that's causing this orange neuron to reach threshold and generate an action potential, but you're getting many excitatory potentials, one after the other, coming to that orange neuron from that green neuron. And the idea is that before that first depolarization has a chance to completely dissipate, the second excitatory input, the second depolarization from that same neuron, follows it right behind, and the two depolarizations add together over time to get us to the threshold to generate an action potential, right? So, and of course, it's a little bit artificial to necessarily separate these two concepts out because you could also have combinations of spatial and temporal summation acting on a neuron. We could have a third panel over there off the screen where it's like, the red and the purple neurons are each sending multiple excitatory inputs, and so you'd have this combination of spatial and temporal summation in that case. Okay, so that's what we have to recognize, right, is that neurons are often receiving multiple inputs, multiple stimuli, either from different neurons at the same time, or the same neuron at different times, or a combination thereof. And it's the summation of all the gradient potentials that occur as a result of those stimuli here that has to get us to the threshold or not to determine whether we generate an action potential or not. On the left-hand side here, they're talking about this idea of cancellation. So here they are using this term EPSP, IPSP cancellation. So as it says in the top there, these stand for excitatory and inhibitory postsynaptic potentials. So what they're trying to show us here is now we've got a red neuron, the same one as over here presumably, but now we've got this blue neuron that's inhibitory, right? So it's going to be, say, releasing some neurotransmitter that opens up a potassium channel on that orange neuron. So we're going to get a hyperpolarization now, an inhibitory input, if you will. And so they're again trying to show us that, the red neuron excited us, got us closer to threshold, didn't get us to threshold, but got us closer. But then the blue neuron's hyperpolarizing effect here just offset the depolarizing effect of that red neuron, and so the excitatory signal and the inhibitory signal, they cancel each other out. It's basically like when you're adding a positive number plus some negative number, right? And ultimately you get yourself back to 0, right? So, so again, what we can appreciate here is that we can have, if we think about the totality of this figure, we can have multiple inputs coming to a neuron from different neurons, from the same neuron over time. Those inputs could be of an excitatory or an inhibitory nature, and the idea is that at the axon hillock, these are all just being summed together, and if they sum together to get us to threshold, an action potential occurs, and if they sum together and we're below the threshold, then no action potential occurs. Okay, the next thing I want to talk about here is what happens to the sensitivity of our nervous system when we experience changes in the ion concentration of our body fluids. Okay? So we talked last week about this idea that, of course, we have dietary inputs of ions. We have our kidney trying to regulate the levels of ions in our extracellular fluid. And so based on based on what we're eating, based on the function of our kidneys, we could experience changes to the concentration of our body fluids over time, particularly to the extracellular fluids, okay? Because that's where all these dietary inputs are coming in. It's our blood plasma that our kidney is trying to, regulate its ion composition. So we can expect to experience possibly over time, especially if our diet is wacky or if our kidneys aren't quite functioning right, we could expect to see changes in especially the extracellular fluid ion concentration over time. And as we're going to talk about next, that can lead to changes in the sensitivity of our nervous system. So let's just look at this figure here. And so we'll just start here with what's basically the normal situation, this black line here. So the first thing to point out here is we've got our resting membrane potential marked here. And then this line up here represents the threshold potential. And the first thing that we have to appreciate here is that the difference in magnitude, or I should say the magnitude of the difference between your resting and your threshold potential, that's what determines how sensitive your nervous system is going to be to stimuli. Right? What I mean by that is that, here's our resting potential. Here's the threshold potential we have to get to generate an action potential. We have to have a graded potential of this magnitude to get us to generate an action potential, which means that a stimuli of that same magnitude is what's required to get us to generate an action potential and respond, therefore, to that stimulus. So that's the first thing we have to appreciate, okay? The difference between the resting potential and the threshold potential in a given neuron, the magnitude of that difference correlates to how sensitive to stimuli that neuron is. Now, what they're talking about next here is a condition called hypokalemia. So hypokalemia, and let me be clear about this definition, hypokalemia means that you have a lower concentration of potassium in your extracellular fluids than normal, okay? So we start off by recognizing, as we did the other day, there's not a lot of potassium in our extracellular fluids to begin with, okay? We only had maybe, 5-10 millimolar of potassium in our extracellular fluids as we were going through the math last week. Hypokalemia means that we're down from that, right? That maybe we're at 1 or 2 or 3 millimolar for potassium. We're at some lower potassium concentration in the extracellular fluids compared to normal. Now, what is going to be the consequence of hypokalemia for the resting membrane potential? Well, if you've got a lower concentration of potassium in your extracellular fluids than normal, because we have a high concentration of potassium in our intracellular fluids, that lower concentration of potassium in the extracellular fluids is going to mean a bigger potassium concentration gradient than normal. And that's going to mean that the equilibrium potential for potassium is going to be more negative than normal, You're going to need a bigger electrical gradient to oppose that bigger concentration gradient. And now, if the equilibrium potential for potassium is more negative than normal, especially given that potassium is typically the ion that our membranes is most permeable to, and therefore it has the biggest say in terms of our resting potential, it's not surprising that our resting potential in hypokalemia becomes more negative than normal. And the consequence of that is that if you look at now the distance between our resting potential and the threshold, it's a much larger difference, which means we're going to need a much bigger gradient potential, and by extension, a much bigger stimulus, much stronger stimulus, to get an action potential. So what does hypokalemia do, as it says here? Well, it doesn't really say it the way I want it to, but it hyperpolarizes tissues, which means that ultimately it makes the nervous system less responsive. So hypokalemia is leading to hyporesponsiveness of our nervous system because it's causing the resting membrane potential to become more negative, moving it further away from the threshold, meaning a stronger stimulus is now required to get to threshold and generate an action potential, to generate a response to that stimulus. If we look at the opposite, which is hyperkalemia, and again, let's define this. So hyperkalemia means that you have a higher concentration of potassium in your extracellular fluids than normal. And what I really want to drive home here about hyperkalemia is, again, we're not saying that your concentration is above the intracellular concentration, right? Again, we only have maybe 5 or 10 millimolar of potassium in our extracellular fluids. Even if we double that up to 10 or 20 millimolar of potassium in our extracellular fluids, that is still substantially below the intracellular potassium level. Okay, the biggest mistake people make is forgetting that hyperkalemia just means higher than normal potassium levels in the extracellular fluid. If you get it confused in your mind thinking it's higher than the intracellular fluid, you're going to be in trouble, right? It's higher than normal levels of potassium in the extracellular fluid. So what's the consequence of hyperkalemia? Well, we still have a potassium gradient that favors potassium leaving the cell. It's just now the magnitude of that concentration gradient has become lesser. And so potassium's not going to leave as easily, right? And so essentially we're going to have more of this positively charged potassium staying in the cell. You know, the other way to think about it is that the electrical gradient, the equilibrium potential for potassium, won't be quite as big a negative now, because we don't need as big of an electrical gradient to oppose this smaller potassium gradient. And so ultimately we should expect, and they show it here, that the resting potential will now be some more positive value than normal. And when you have a resting potential that's more positive than normal, and therefore, as they depict here, closer to the threshold potential, then that's going to mean that the nervous system becomes more responsive to stimuli. Because now only a small gradient potential, and by extension only a small stimulus, is required to get us to the threshold potential to cause an action potential. So hyperkalemia is going to lead to hyper-responsiveness of the nervous system. So this is the thing we have to keep in mind, right? If we change the concentrations of ions, and we're focused here on the extracellular fluid, because again, that's usually where we see the changes occurring. They could be occurring intracellularly as well, and then you still have to just go through the same thought process to figure out, well, what's going to happen here? But we can appreciate that by having our basic understanding of the resting membrane potential that we developed last week, we can say, hey, if we change the concentration of an ion in the extracellular or even the intracellular body fluids, we can work out from those basic principles last week, how's that going to affect the resting membrane potential? Does it move it further away or closer to the threshold? And based on that change, we can predict whether the nervous system becomes more or less responsive to stimuli. Okay? So that's the thought process there. Now, there is one additional piece of information that I want to throw at you here, because it ties nicely into some stuff we're going to talk about either later today or Thursday. It says here that if the potassium concentration in your extracellular fluid gets really high, right, so it goes way, way above normal, right, still could be below the intracellular, but let's go up to like 50, 60, 70 millimolar, let's say, the amount of depolarization that you get could be so great that it actually sits now above the threshold potential. What happens in that case where your potassium is so high that the resting membrane potential is above the threshold potential is your nervous system shuts down. And it shuts down, and this will be clearer as we talk about action potentials in a minute, it shuts down mostly because the voltage-gated sodium channels needed to generate action potentials get completely locked in an inactivated state. Okay, I'll come back to this point when we get to talking about those channels in a bit. But there is kind of this interesting relationship where initially hyperkalemia makes your nervous system more responsive, but it gets to a point where it actually shuts the entire nervous system down. All right? And so again, we'll come back to this point a little bit later when we start talking about action potentials. Yeah, and I put this sign, I guess I put this up here just to talk about symptoms of these things. But yeah, if you're thinking about hypokalemia, for example, I guess I meant to say that, you know, your nervous system's becoming less responsive, right, in hypokalemia because your membrane potential is more negative than normal. One of the major symptoms of hypokalemia is people notice muscle weakness, right? They notice muscle weakness mostly because it's becoming harder to stimulate the muscles to contract because their resting potential is becoming more negative, so it's harder to stimulate them to contract, so people often notice weakness. The interesting thing, too, is that you get the same symptom with hyperkalemia once you get to that point where the hyperkalemia is so severe as to be above the threshold potential. Because again, now suddenly you can't really stimulate your muscles to contract because your nervous system is kind of shutting down, and so people often notice weakness with severe hyperkalemia as well. Okay? So anyways, just kind of trying to scale this up, I guess, to what you see at the level of the whole person. Okay, next up I want to spend a little time talking about a very special type of graded potential, something called the pacemaker potential, which we see in certain specialized cells. The pacemaker potential, one of the places where we see it, and where we're going to kind of talk about it here, is the pacemaker cells of your heart, right? So you've got pacemaker cells in your heart that are helping to generate an endogenous heartbeat, okay, without any requirement for any kind of stimulus, right? So your heart is able to generate its own endogenous heartbeat without any need for nervous system input or any other sensory input. This is part of the reason why even after you're dead, and maybe we take your heart out, I mean, it'll continue to beat for some time thereafter, right? Just because it has this endogenous pacemaker built in. Now, the defining feature of a pacemaker potential, as they highlight here in this figure, is that it never reaches a steady state, right? So the pacemaker potential here, it doesn't stay steady at some resting potential value like we typically see. It's characterized by this slow depolarization. So here you can see, for example, we had an action potential occur, and let's imagine that action potential causes your heart to beat, right? And then after the action potential, you know, we see this pacemaker potential slowly depolarizing us back to the threshold, so we get another action potential, the heart beats again, and that keeps happening over and over. So because of this non-stable, because of this slow depolarization that characterizes the pacemaker potential, there are action potentials being generated to stimulate the heart musculature, you know, on some routine basis here, without there having to be any stimulus being sent to the heart. So that's the nature of a pacemaker potential. It's characterized by this unsteady, slow depolarization to threshold, allowing for the spontaneous generation of action potentials over time, again, without any kind of stimulus input. So the question to ask here is basically, well, what is underlying the pacemaker potential, right? We know that membrane potentials tend to relate to channels, and we've talked about leak channels, and we've talked about gated channels. So what kind of channel is at play here? the channel that is responsible for pacemaker potentials is something called the HCN channel. And HCN stands for hyperpolarization activated, so that's the H, cyclic nucleotide gated, that's the C, sodium channel, which is the N, right? So those are the three defining features of the HCN channel. They are activated by hyperpolarization, they are gated by cyclic nucleotides, and they are sodium channels, all right? So the figure here is showing us an HCN channel, and so you can see when it talks about cyclic nucleotide gated, you can see what they mean here. So they're talking specifically about cyclic AMP, right? And so the idea here is that basically when you have cyclic AMP bound to this channel, it becomes more permeable to sodium. So even in the absence of cyclic nucleotide, you will get some movement of sodium through the channel, but as you have more and more cyclic AMP bound to the channel becomes more and more permeable to sodium, so you get more and more sodium flow through the channel as you have more and more cyclic AMP bound to it. In terms of the hyperpolarization activated part, again, I'm putting the cart a little bit before the horse here, but at the end of every action potential, we have what's called an after hyperpolarization. So the membrane potential at the end of every action potential, the membrane potential actually goes down below the resting potential for a period of time. We call that an after hyperpolarization. That after hyperpolarization is what activates this HCN channel so that it starts to open and allow for the flow of sodium to pass through it. And so this is what allows then for this pacemaker function, because it's basically an action potential, specifically the after hyperpolarization of the action potential, that is triggering this channel to become active, to start letting sodium flow through it, which then explains why we get this slow depolarization after each action potential that gets us back eventually to threshold, so another action potential can occur, and then the after hyperpolarization of that action potential opens the channel to let sodium go through and so forth. And then all we're saying is that the cyclic AMP here kind of controls how easily the sodium can pass through. The more cyclic AMP bound to it, the more that sodium passes through. The more sodium that can pass through, the quicker we're going to depolarize back to threshold. So you can imagine that cyclic AMP is helping, for example, to speed up the heart rate here. And if you had low levels of cyclic AMP, it'd be the opposite. There wouldn't be a lot of sodium flow through the channel, so the depolarization would take longer and longer to get to threshold, and that would slow down the heart rate. Okay? Indeed, actually, maybe I'll come back to this point in a second, but indeed, this figure that I have here is going to show us how this HCN channel, which then controls heart rate, we're going to see here how this ties into our earlier conversation about the sympathetic and parasympathetic nervous systems, which we know can, slow down and speed up the heart rate. So here's our HCN channel in this diagram, right, allowing sodium to pass through. Here in the membrane, we have this enzyme called adenylyl cyclase, and this is an enzyme that converts ATP into cyclic AMP. So it's our cyclic nucleotide generator. And what you can see here, if we look at adrenaline first, so adrenaline is just sort of the older name, shall we say, for epinephrine, right? So we have adrenaline and noradrenaline. They're the same thing as epinephrine and norepinephrine. We already know that epinephrine and norepinephrine, these are the major neurotransmitters of the sympathetic nervous system. And we know that the sympathetic nervous system speeds up our heart rate. So here what we see is the adrenaline or epinephrine binding to its corresponding receptor, something called an adrenergic receptor. We're going to talk way more about these receptors later in the course. And we can see how the binding of epinephrine to its receptor activates the adenylyl cyclase. So the adenylyl cyclase becomes more active now, right? And so it starts to produce more cyclic AMP, and then that cyclic AMP binds to the HCN, making it more permeable to sodium. So you're going to have more sodium coming through this channel at a faster rate, and that's going to lead to a quicker depolarization to threshold, and therefore a faster heartbeat. And then acetylcholine has the opposite effect here. So acetylcholine, we know it to be the major neurotransmitter of the parasympathetic nervous system. So here we have a receptor for acetylcholine. It's specifically called a muscarinic receptor. And again, we're going to talk lots more about this receptor later in the course. But when acetylcholine binds to the muscarinic receptor, it has the opposite effect. It inhibits the adenylate cyclase. And therefore, you're not going to be able to generate generate cyclic AMP as rapidly now. So you're going to end up with lower concentrations of cyclic AMP. There won't be now as much cyclic AMP binding to the HCN channel, so sodium won't flow through that channel quite as quickly, and that means that the depolarization to threshold will take longer, and so now you'll get fewer action potentials per unit time, and consequently a slower heart rate. So again, just kind of tying this HCN channel and its pacemaker function back to what we know about the parasympathetic and sympathetic nervous systems. Let me just go back for a second here to this little figure that I had here, because what I wanted to mention with that figure is, basically this HCN channel is a voltage-sensitive channel then. And in just a bit, we're going to talk about some other important voltage-sensitive channels, the ones that are necessary for action potentials, like, for example, the voltage-gated sodium channel and the voltage-gated potassium channels. Later in the course, when we talk about neurotransmitter release, we're going to talk there about voltage-gated calcium channels. So really what this figure is trying to show us is that these HCN channels, they are part of this bigger family of voltage-sensitive ion channels, right? So again, we've got the sodium and the potassium and the calcium voltage-gated channels. These HCNs, they are part of that family, right? So all these voltage-sensitive channels that we have in not only humans, but kind of animals in general, they're all kind of ancestors of a single original voltage-gated ion channel. Okay, I also wanted to just mention here kind of briefly that, of course, we just talked about the HCN channel in the context of the pacemaker in the heart, but I did want to show evidence that these HCN channels also are present within the nervous system. This is a study where they were looking at rat brains, and you can see here, first and foremost, there are various different isoforms of these channels, and in fact, that was, of course, also noted here on the figure as well. There are 4 different isoforms of these channels. And what they're showing here is that these different isoforms of HCN, they are expressed in different brain regions. So where you see kind of the darkest regions on these figures, okay, that it shows you where this particular HCN isoform is being expressed in the brain. So again, we don't have to get too worked up about where these different isoforms are, but the basic point here is there are different isoforms of HCN, and they are kind of each expressed in different brain regions. And we do have some evidence that they have a function, albeit not a clear pacemaker function, so it's not entirely sure why these channels are responsible for what we're going to see here. But let me just introduce you to something called the von Frey test. This is a very common test done to really test neuronal sensitivity in rodents. And so the way this works here is you would have your mouse or your rat, and they're kind of standing here on this wire cage. And then what you do is you have this little kind of probe, if you will, below that's going to push on the foot of that mouse through the cage, right? It's going to gradually apply more and more pressure to the mouse foot, and then you're going to figure out, okay, well, how much pressure can I apply before the mouse detects that and pulls its foot away, right? So you're just trying to measure the nervous system sensitivity of the mouse here by basically pushing harder and harder on the bottom of its foot until such time as it responds, and then you can say, okay, well, here's how much pressure or force I could apply to that mouse foot before it responded to it. So here, they did this von Frey test. So you can see in this case, they're measuring the threshold force in units of grams here, I guess. They're basically measuring like how much weight do I have to kind of apply to the mouse foot here in order to get it to respond. So if you look at the baseline measurement, right, you can see that the mouse shows a response at maybe 2G here, right? So it's pretty sensitive to this force. But then they show that if you infused into this mouse a compound that blocks the HCN channel, right? So you're trying to inhibit this channel, stop this pacemaker potential. Notice that 30 and 60 minutes after you administer this channel blocker, there's almost a 5-fold decrease in the sensitivity, right, of the mouse nervous system. You have to apply 5 times as much force to the mouse foot here to get this mouse to respond now. So again, if you think of the HCN channel as a sodium channel, which it is, and that sodium channels obviously depolarize the membrane, so they bring it closer to threshold, make it easier to excite the nervous system, then these results make perfectly good sense. You're basically blocking a sodium channel. That's going to ultimately lead to the hyperpolarization of the resting potential, and that in turn is going to make the nervous system less responsive to stimuli, which is exactly what we see. As I said, what's not entirely clear from these data, and as far as I can tell, no one's really got a good answer to this question, like, where is the pacemaker part of this whole thing coming in? You've got an HCN channel that we know is creating pacemaker potentials. It's not entirely clear why, nervous system sensitivity to, say, this pressure stimulus needs to have any kind of pacemaker rhythm to it. So that's the part that remains a little bit unclear, but at least we can see that there are HCN channels in various brain regions, and we definitely know that they are having physiological effects in terms of the sensitivity of our nervous system. Okay, so with the last few minutes that we have here, we'll get started thinking about action potentials, and then we'll be able to go into them in a lot more detail on Thursday. So let's just start with the definition here. So it says action potentials are brief, large reversals in membrane potential that can be propagated over a long distance, such as the length of an axon, and they only occur in excitable cells, like neurons. Well, let's just look at that definition a bit here, because I want to contrast it in particular with the definition of graded potentials that I gave last week. So let's start with the idea that they're brief. Action potentials are brief. And you can see what that means if you look at the figure. This action potential is only really lasting for, a couple of milliseconds. So they are brief. And we can contrast that with graded potentials, because we noted in the lecture that graded potentials, their duration is variable depending on how long the stimulus is in place, right? So you could have graded potentials that are, as short as this millisecond length, but they could be, seconds long, really, if you have a stimulus that's being applied for a longer duration. So action potentials, very brief. Graded potentials can be much longer. It notes here that we have this reversal of membrane potential, and what they mean by that is, as we know, the resting potential of a neuron down around -70. At the peak of an action potential, we have a positive membrane potential value, right? So we've seen a 100 millivolt swing in membrane potential from resting value to the peak of that action potential. We've gone from negative to positive membrane potential. Again, that's very different from a graded potential, because graded potentials, I guess in theory they could get you to some positive membrane potential, but you probably wouldn't necessarily see that. I mean, you'd probably get to the threshold potential and have an action potential occur before you'd see such a big graded potential. So we are seeing a reversal of membrane potential here at the peak of the action potential. So they're much bigger, really, in terms of the magnitude of change. And then they can be propagated, self-propagated over a long distance, right? And we noted some axons can be, a meter long, maybe even longer if you're thinking about, bigger animals than us. And again, that's in stark contrast to the decremental conduction that we see in graded potentials, right? We just talked about that this morning. The graded potentials decay and therefore are very localized phenomena, right? Occurring over a couple of millimeters at best, as we saw last week, definitely not able to propagate over meters of length, okay? The one thing that certainly unites them both is they both only happen in excitable cells. So again, we have this notion that the resting membrane potential is a universal cell trait, right? All cells have a resting membrane potential, but that graded potentials and action potentials are unique to excitable cells, which again, by definition, are cells who can change their membrane potential by changing their membrane ion permeability, okay? And actually, maybe as a kind of point to drive that definition home, we talked a little bit earlier today about how membrane potentials of cells can change in response to changing ion concentrations in body fluids. If you had a cell whose membrane potential changed in response to changing concentrations of ions, you would not be able to say that was an excitable cell. Because again, the definition of an excitable cell is I can change my membrane potential by changing my membrane ion permeability, which is going to be accomplished through changing channels, right? Having more or fewer or open or closed natures of channels. Action potentials are also known as nerve impulses. So you'll see that term used, especially perhaps in older scientific literature. Action potentials depend upon voltage-gated channels, which we're going to talk about on Thursday. So you can see pictured here, voltage-gated sodium and voltage-gated potassium channels. Both are key to the action potential generation. And then lastly, voltage-gated channels are, for the most part, found exclusively in the axon initial segment or the axon. Now I say for the most part here because we certainly expect to see voltage-gated sodium channels in those regions because that's where the action potentials are generated and propagated. The for the most part refers to the fact that we do in some neurons find voltage-gated sodium channels in the dendritic zone. In the dendrites of the cell body, what they do there is they're like signal boosters. Because if you have decaying graded potentials, but you need to somehow boost that signal so it can travel further, that's where these voltage-gated sodium channels come in. So they're kind of like, I was thinking like, this weekend how often my Wi-Fi drops out when I'm in the middle of video calls and I have to get maybe some signal boosters to kind of, boost the signal as it gets too far away from the router. The voltage-gated channels in the dendritic zone are that exact function. That you have a stimulus that occurred here and it decayed on its way to the axon hillock. If you want to be able to boost that signal to get it to be stronger when it gets to the hillock, you can actually have voltage-gated sodium channels in the dendritic zone that will serve as that signal booster. Okay, so we'll wrap it up there, and we'll talk Thursday all things action potential, and I'll see you then. It could be either way. So basically we're saying you've got a stimulus that has occurred here, so a graded potential formed. That graded potential is decaying in magnitude as it travels. It might therefore be subthreshold by the time it gets to the axon initial segment. If you want to boost that signal to kind of oppose the decay and make it strong enough to actually still create a stimulus at the axon initial segment, that's what you can use those voltage-gated sodium channels to do, to kind of boost the magnitude of that signal as it's traveling through the dendritic zone. And that would occur in either of these two circumstances, right? Because ultimately, whether you have these two neurons causing an excitation here, and then you want that excitation to get to the axon hillock to create the action potential, you can have those voltage-gated sodium channels in that orange neuron to boost the signal timing from either or both of those neurons, or to boost either of the signals coming from this neuron here. So it doesn't matter. Yeah.
And then these like summations, translations, they don't do they guard specific neurons or just like
It's a general. Yeah.
just general?
It's a general property of neurons. Yeah. Okay. Okay. All right. What's up? Yeah, for sure. Well, I mean, and you were the first one that went, right? Yeah, yeah. Because, which was about the. Yeah, yeah. I mean, so it's an intriguing idea, right? I mean, like I said, it's hard to differentiate between all these ideas because they're all kind of the same.
Yeah.
You're all working on two kinds of cancers, and you're all trying to figure out like these non-invasive screening tools. So they're all so similar. Yeah, the only thing I got a little bit confused about here is the only thing I think could really improve is, again, how you're getting those samples in terms of like, because at one point you seem to imply like, okay, the cells have to kind of be broken up in order to release the signal. Later you seem to imply, well, but there's also going to be just like this naturally happening, and so it'll just be in the fluid and I can suction it up. So it just felt like the details were a little bit unclear, and so that made it hard for me to kind of follow what you were talking about at some point. And it makes me unsure that you know what you're talking about. So that's really the only thing I would say. We'll walk and talk here, so we'll pull it on. Yeah, I think that was the part that just...